Hi, I'm Indu. I'm a postdoc in Professor Howard Griffith's lab at the Department of Plant Sciences at University of Cambridge. My research focuses on algal carbon concentration mechanism. The title of my talk is Microbial Carbon Concentration Mechanism, a toolkit to enhance agricultural production. So during the course of the talk, the first few minutes, I shall focus on introducing the topic of carbon concentration mechanism, and then we'll go on to explain why we study it and how it could act as a tool to enhance agricultural production. So what is the basis of all life on Earth? And the answer to that question is photosynthesis. And what is photosynthesis? It is the process through which inorganic carbon in the form of carbon dioxide is utilized by photosynthetic organisms, and they convert carbon dioxide to usable forms such as glucose. And photosynthesis is the only process on the face of the earth that converts carbon dioxide to a form usable by living organisms. The glucose so formed can then be introduced into various pathways to form the other biomolecules required by living organisms. And all life on Earth is carbon-based. So this is a pivotal process to sustain life. Now the question is, who photosynthesize? Well, pretty much anything and everything that appears green around us photosynthesizes. Trees, shrubs, herbs, what you have, all of them photosynthesize. But in addition to all these plants and trees that we see, there are also microbes which live in water bodies that photosynthesize. And their contribution to fixation of carbon dioxide is pretty substantial. To illustrate, if one were to compare the biomass of aquatic microbes to that of terrestrial plants, they constitute about 1/20th of terrestrial plants. However, when one compares their annual net primary productivity in terms of how much carbon they manage to fix, their contribution is almost equal to that of terrestrial plants, which is a pretty incredible feat considering the size, considering their sizes. So how do aquatic microbes manage to punch about their weight? And here we'll come to another important molecule, rubisco. Rubisco is one of the most important enzymes on Earth. And why is it? It's a key enzyme which, in the process of photosynthesis, which catalyzes a reaction that involves combining carbon dioxide with a 5-carbon molecule called ribulose bisphosphate. And it is absolutely pivotal for the process of carbon fixation. However, rubisco has issues. Rubisco evolved about 2 billion years ago in an environment which is carbon dioxide rich. However, owing to photosynthesis, over millions of years, our atmosphere has become oxygen rich. Rubisco is primarily carrying out the reaction of carbon fixation. However, it can also carry out a promiscuous reaction with oxygen, leading to a process called photorespiration, which leads to loss of fixed carbon. And the issue is compounded by the fact that there is more oxygen than carbon dioxide. And this is further compounded in the case of aquatic microbes and plants, because in water, the availability of carbon dioxide is even lesser as opposed to air. So how do aquatic microbes handle this situation? Well, they've evolved mechanisms, and this particular mechanism is called carbon concentration mechanism. And this is a solution for rubisco's promiscuity. So what happens in carbon concentration mechanism? Essentially, there's a concentration of carbon dioxide within the cells of the organism, and it is delivered to a concentrated mass of rubisco, thereby reducing rubisco's exposure to oxygen. And by providing more carbon dioxide to a concentrated mass of the catalyst, we ensure that there is more of the carbon fixation happening, and it is happening more efficiently. So where does carbon concentration mechanism primarily occur? But as I mentioned, it happens in several of the aquatic microbes. And some of the most well-studied carbon concentration mechanism organisms are cyanobacterial species and green algae. Shown on the left are the microcompartments where in cyanobacterial species, and these dark blobs are the microcompartments where rubisco is held concentrated, and this is where the concentrated carbon dioxide is delivered. On the right is a EM image of Chlamydomonas reinhardti, the model organism that I study. It's a unicellular green alga. And the arrow points towards a pyronoid, the organelle, which is essentially a concentrated mass of rubisco, which is where the carbon dioxide is delivered. And this concentrated mass is called pyronoid, which is the seat of carbon concentration mechanism in the green alga. So what exactly happens in green alga? So the green alga, as I mentioned, is an aquatic microbe. It's unicellular. And in water, carbon dioxide, the dissolved carbon dioxide, exists not just as carbon dioxide, but in ionic forms of bicarbonate and carbonate. And the ratios of the ionic forms as opposed to carbon dioxide depends on physical factors such as temperature and pH. There are transporters to transport these inorganic carbon forms, both bicarbonate ions and carbon dioxide, into the cell, into the chloroplast, and finally to the pyronoid. And in the pyronoid is rubisco, and also carbonic anhydrase molecules, which will convert bicarbonate to carbon dioxide, because rubisco can only act on carbon dioxide. So why are we studying this process? Well, if we could engineer something similar to the algal CCM into higher plants, then we would technically be making them more photosynthetically efficient and thereby would increase their yields. But to do this, we have to understand this process very well within the organism itself. Further, it is a process which is over 400 million years old, and anything that has survived these many years of evolution merits research and examination. So how do we go about engineering algal CCM into higher plants? Well, we'd have to identify the key components in the pyronoid and in Malden CCM, characterize them, and then figure out which components to transfer to higher plants, genetically modify higher plants, introduce the key components, and then ensure and see what needs to be regulated to have CCM functioning within the higher plants. So through the CAP consortium, of which Harvard has been part of, we have collaborators at Princeton, led by Martin Yonikas, at Edinburgh, led by McCormick, at University of York, led by Luke Mackinder, who have been trying to engineer plants with CCM. And they have made some progress in introducing the pyronoid-like structure in higher plants. And they've done this in the model organism, Arabidopsis. So what they have been able to do is to mimic the microcompartment, which does not exist in higher plants. And they've been able to introduce this in Arabidopsis by introducing algal components. However, this pyronoid-like structure still needs to be integrated with the other cellular components to have a functional CCM. So there is some way to go, but there has been substantial progress. So what do we do in our lab? Well, we are studying various aspects of algal CCM, which include the following projects. We're looking at how the day-night cycle is involved in regulation of algal CCM. We are also studying novel CCM components that have been identified computationally, and we are looking at their role in CCM. We have looked extensively at the role of cells' protein degradation machinery, and we are also looking at identification of cis-regulatory elements important in algal CCM. So shown here, I have some snapshots from the experiments, the kind of experiments that we carry out in lab. So here, what I have is a Western blot of a gel wherein we have this key protein trapped in its different phosphorylated forms. And we see that over the course of the 24-hour cycle, there is a dramatic change in the extent of phosphorylation of this particular protein. And this is a key component of pyrimide, indicating how this particular protein has to be regulated to have the CCM going in the algae. Shown on the right is a spot test where we are screening for multiple algal knockouts to see which genes might be important or have a role in CCM. We have identified here, what I have here is are two knockouts, which I have labeled with red asterisks, which seem to do much better than the wild type, seem to have a far more efficient CCM as opposed to wild type. And we are in the process of characterizing these regions in collaboration with University of Essex, and we are hoping to see what we get out of it. The role of protein degradation machinery is being explored by mass spectrometric analysis, and we are looking at cis-regulatory elements in algal CCM, which again hasn't been characterized previously through ATAC sequencing. With that, I would like to acknowledge the various people involved in this project. Rachel is an MPhil student who recently submitted her master's thesis and got her degree, worked on the EPIC1 phosphorylation story, and is also currently involved in looking at the degradation story in the context of CCM. Tanya was an exchange student who began the degradation story along with me. Ella Catherall was an undergraduate project student who looked at red algal plastid proteomes. Alice, Sophie, and Priyanka worked as summer interns in the lab, and Priyanka was pivotal in characterizing several knockouts during this summer, which we are pursuing further. Dr. Geetanjali's group and her students, C2 and Archana from NIPGR, have been collaborating with us, and they've been carrying out computational analysis and network analysis of the CCM components. And I'd like to thank Professor Howard for having given me the opportunity to work here, and all the inspiration and good energy that he's provided us with in the lab. Thank you.